Good morning. Today I will discuss local energy decomposition methods in ORCA. These are tools that can be used to obtain chemical insights into the nature and properties of chemical interactions in molecular systems. This includes systems with hundreds of atoms and complex electronic structure. The local energy decomposition scheme is available for a broad range of local correlation methods, including HFLD, the LPNO couple cluster with singles and doubles, and the LPNO CCSD parenthesis T. Each of these schemes has a different accuracy and efficiency. For very large systems, these methods can also be combined using the multi-level methodology. We can, for example, define different fragments in a system and then treat each fragment and the interaction between the fragments at different levels of theory. So we really have a plethora of tools that allow us to tackle different situations, each time focusing the computational efforts in the part of the system that is of interest to us. In particular, the DLPNO couple cluster methodology is a local correlation variant of CCSD parenthesis T, which enables the calculation of couple cluster energies and properties for systems with hundreds of atoms. This approach reduces the inherent steep scaling of the gold standard CCSD parenthesis T method to nearly linear, and therefore has found widespread applications across many fields of chemical research. We frequently use it in our own research, especially when dealing with systems where it is unclear which exchange correlation function might be best suited for a given application. One especially relevant application of the DLPNO CCSD parenthesis T method concerns the accurate modeling of binding processes. For example, we can use this approach to quantify accurately the energy required to bring together water molecules and form a water cluster, or to compute lattice energies of molecular solids, protein ligand binding energies, or even the energy released when two strands of DNA come together to form a double helix. Now, in the context of the DLPNO couple cluster method, this is typically done using a supramolecular approach. So the only thing that is needed is to perform a series of single-point energy calculations for the molecular adduct and for its constituting fragments. For example, for computing the association energy between a ligand and a protein within the supramolecular approach, we need to compute the energy of the protein ligand adduct and then subtract to this energy the energy of the unbound ligand and of the original protein. And after having computed the binding energy, again, for example, at the DLPNO CCSD parenthesis T level, the local energy decomposition scheme allows us to break it down into contributions from individual chemical interactions. For example, the interaction between a residue in the active site of the task of the protein and the ligand. Now, this scheme has found widespread applications in the study of non-covalent interactions, but before diving into the key aspects of this feature, I will briefly recap the main idea behind this approach with a few examples. So, for instance, consider again our example of the interaction of a protein with a ligand. With the local energy decomposition, we first divide the system into fragments, such as the ligand and the key residues in the active site of the protein. We then compute the binding energy at the DLPNO couple cluster level between the ligand and the protein using a supramolecular approach, and then using LED, we decompose it into contributions from individual fragments and fragment pairs. In this decomposition, you will see one repulsive contribution per fragment, representing the electronic preparation, and this is the change in the electronic energy of the fragment from its ground state to the perturbed state it has in the protein ligand complex. Additionally, we have the interfragment contributions, and these represent genuine pairwise interactions. These can be further decomposed into electrostatics, exchange, and dispersion, thus yielding additional insights. So how does the LED work? In this approach, we exploit the fact that the electronic energy in any post-Hartree-Fock method can always be expressed as the sum of contributions from at most pairs of particles, electrons and nuclei. By assigning each particle to the fragment in which it is dominantly localized, it is straightforward to divide the energy into intrafragment contributions, where all particles belong to the same fragment, and interfragment contributions, where the particles belong to different fragments. The definition of interfragment electrostatics, collecting all the potential terms, and exchange is also, of course, straightforward in this approach. It is important to note here that molecular orbital assignment into fragments is trivial because we are in the context of local correlation approaches. And these methods employ localized orbitals, which are orbitals that are confined within a limited regional space. Here you see as an example the Foster-Boyce localized orbitals of the water dimer, which can be readily assigned to each of the fragments. The decomposition of the correlation energy instead is slightly more complex because it is based on the localization of both the occupied and the virtual orbital space. This allows the decomposition of the correlation energy into families with well-defined physical meaning, including, for example, the dispersion excitations, which, as you know, are responsible for the attractive part of the van der Waals potential. Now, for systems with many fragments, the results from the LED analysis are usually represented graphically by means of the local energy decomposition interaction maps. Here is an example of these maps for the decomposition of the binding energy of the water hexamer. Now, in this matrix, the diagonal terms represent the repulsive electronic preparation associated with the change in the electronic structure of water molecules upon formation of the cluster. For example, the electronic preparation of water 2 amounts to 41.1 kilocal per mole. In contrast, the off-diagonal terms represent genuine fragment pairwise interactions. For example, the strongest fragment pairwise interaction in this complex occurs between water 5 and water 6 and amounts to minus 72.6 kilocal per mole. This is a significant negative, so attractive contribution to the binding energy, and represents all the electrostatics, exchange, and dispersion interactions between these two water molecules. Now, using LED, these interactions can be quantified individually to obtain additional physical insights. So this is the standard LED approach, and this approach separates, as I mentioned, the repulsive and attractive contributions to the binding energy. Therefore, it is important to note that interfragment terms obtained in this way do not include the main repulsive contribution associated with the binding process, making them relatively large compared, for example, with experimental water dimer binding energy that amounts to about 5 kilocal per mole. So obtained results that align more closely with the experimental results, the electronic preparation terms that are represented here in the diagonal must be redistributed across all fragment pairwise contributions. This allows us to estimate the net interaction effects and achieve values more comparable with experiments. The approach that makes this possible is called fragment pairwise local energy decomposition. With this approach, we no longer have the diagonal terms because they have been distributed among all fragment pairwise interactions, but we have instead an exact pairwise decomposition of the association energy of the water cluster. For example, the strongest interaction between water 5 and water 6, now accounting for all repulsive and attractive terms, amounts to minus 9.6 kilocal per mole. This is roughly twice the interaction for the isolated water dimer, and this difference originates from the cooperativity of non-covalent interactions within the water cluster. In contrast, if you look at the interaction between water 5 and water 3, this is just 4.2 kilocal per mole, meaning that in this case, the interaction is weaker than that in the isolated water dimer. And this effect originates from geometric perturbation effects associated with the formation of the water hexamer. So overall, this approach provides results that can be directly compared with experimental data, offering quantitative insights into the relative strength of individual interactions and cooperativity effects. It is also important to note that by comparing the results obtained from the fragment pairwise local energy decomposition map with those from a simple two-body approximation, it is possible to develop cooperativity maps, quantifying precisely the impact of cooperativity of different non-covalent interactions. If you're interested in learning more about how to perform this kind of analysis, you can have a look at the paper report here. So finally, if you would like to know more about the LED scheme in general, including the basic principles and the implementation aspects and applications, you can have a look at the references reported here. And with this, I would like to thank you for your kind attention.